Next one up is Sara Busatto. She's in Jerry Wolfram's lab, and slightly switching gears now, going to the biosensor applications of extracellular vesicles.
So good morning, everyone. Thank you to be here. It's my first year at CLINAM, and I have to thank you, the organization, because it's a great conference to meet and discuss great science with great people, so I'm really happy to be here. I am going to talk about our extracellular vesicle-made supported lipid bilayer. So, previous speaker already talked about extracellular vesicles, their characteristic, their originating pathways. So, the concept for my talk is that they are really important in intracellular communication. Specifically, they mediate both pathological, physiological, and also pathological processes. For example, it has been demonstrated that they are related to premetastatic NIC formation. For example, the pancreatic tumor cells produce vesicles that prepare the liver to receive metastatic cells. And if we move into another pathogenic process, for example, the atherosclerotic plaque, the vesicles produced from coronary artery smooth muscle cells, together with collagen, form this microcalcification that destabilize the atherosclerotic plaque, promoting acute cardiac event. So since they are really involved into pathological processes, they started to be envisioned as a target of therapy themselves. We wonder if in the future we will be able to selectively inhibit the extracellular vesicle biogenesis and release of specific cells, or if we will be able to remove specific extracellular vesicle population from the circulation of the patient, or again, if we will inhibit the extracellular vesicle uptake by recipient cells. So since they are envisioned as a target of therapy, we need to know as much information as possible about their membrane properties, and also to study the interaction of drugs and molecules with their surface. For that reason, we thought that was interesting to develop a supported lipid bilayer, which is the first step to build an extracellular vesicle-made biosensor, since pharmacology use biosensor for drug discovery and drug analysis. When we make a biosensor, we have to be really sure that our layer is immobilized onto a surface, and then during the processing, we do not compromise the biocomponent of our layer and also their integrity or activity. So we started isolating extracellular vesicles from serum-free cell culture media. We isolated them from a prostatic cancer cell line. We checked their biomarker expression, their morphology, and their size. And after that, we were pretty confident that our extracellular population matched the criteria established by the International Society of Extracellular Vesicles. We moved further, and we synthesized the supported lipid bilayer. As a control, we also check the supported lipid bilayer made of synthetic liposome, because we perfectly knew how it was made and the mechanism. So from this slide has a lot of data that can be discussed, but I will go briefly saying that we realized that the supported lipid bilayer was formed through crowding, rupture, and fusion pathways, and that differently from the synthetic made supported lipid bilayer, our extracellular vesicle made require a lot more time after the washing with milky water to open up and stabilize onto the glass surface. And if we look at the fluorescent correlated spectroscopy data, we see that there is a fast decay in the extracellular vesicle-made supported lipid bilayer, because a lot of the probe is washed away at the beginning, maybe because the vesicles are also made by protein, so they cannot acquire as much probe as a liposome can do. But the last part of the cure, or of the curve, quite resemble the one of the liposome-made supported lipid bilayer. And then we have a confocal microscopy analysis where we labeled both the bilayer with the two lipophilic probe. The first one, the green, is PolyPi and is more show more affinity to the fluid pattern of the bilayer, whereas the DIL-C20 show more affinity for the liquid pattern of the lipid bilayer, showing that the extracellular vesicle-made supported lipid bilayer has these red island-shaped areas. That means that we were able to transfer also the lipid raft onto our supported lipid bilayer structure. Then next, we also checked the morphology of the supported lipid bilayer in the middle of its formation with AFM. This is a topography image. And where the supported lipid bilayer is already formed, we have a thickness around 6 nanometers, whereas where the whole EVs is analyzed, the thickness is higher because we have still a sphere and not a bilayer. We also did, we analyzed again with the AFM, doing some punch-through events using the AFM tip of our supported lipid bilayer. And as you can see between the comparison with the liposome-made supported lipid bilayer, the thickness of the bilayer is the same. Here you see the first punch-through event with the first layer and then the second layer, and here again, first layer and second layer. The main difference is that here the tip experienced a higher force because it touched the bottom glass, whereas in the case of extracellular vesicle-made supported lipid bilayer, it seems that the bottom is more soft. So after all this analysis, we concluded that we were able to make a supported lipid bilayer, but specifically our vesicles open into two different ways. The first way, the outer layer of the vesicles attached to the glass surface, whereas in the other case, we call this mechanism parachute type, because the vesicles open and entrapped his cargo in between the bilayer and the glass. So it's for that reason that sometimes we realize that softness in between. We wanted also to be sure that after the synthesis, we still have some molecules which were characteristic of our extracellular vesicle population. So with the dot plot experiments, we were able to determine that C-Sark protein, that is a protein not only related to the vesicle formulation specifically of our cell line, but is also cancer-related protein. It was exposed, released through extracellular vesicles and exposed into the outer layer of the extracellular vesicle structure. So we perform an immunoassay using a green fluorescent labeled antibody against C-Sark, and we compare, we did the immunoassay on both liposome-made and extracellular vesicle-made supported lipid bilayer. And as you can see here, the protein is still able to be recognized by its antibodies, so we preserve the presence of the extracellular vesicle typical protein onto the supported lipid bilayer. So this is just a proof of concept study, and this is our data. And I would like to thank you for the attention, and please, if you have any question, thank you so much.
Very interesting. Have you tried to pre-treat your glass in any way and play with the preparation of the substrate before you started making your film?
No, this is an idea. We are still working on further processes with this project, and so far we just do the supported lipid bilayer in the just cover slip, so just regular glass without any pretreatment.
It may make a difference.
Yeah.
Thank you very much for your great presentation. It was very
Thank you.
interesting. Maybe I missed that at the beginning. So you want to use this layer like a sensor. So how are you going to observe I imagine that you want to do this with a connection with some receptors,
Exactly.
to binary receptors, but what are the techniques that you want to use it to detect that? You want to use this in real-time detection, for example, or you want to use AFM, confocal imaging?
We are talking now with the Tallahassee University in Florida to couple this technique with SPR to then perform some binding experiments with the antibodies or molecules.
Okay, great. Thank you very much. But then I'll have a talk with you later.
Okay, thank you.
I think what was also intriguing was the fact that Argonaute-2 on your dot plot could not be detected if you do not add tween. Is that correct? Did I see that correctly?
So
Is that
the presence or absence of detergent in the dot plot is
Yes.
because if you don't add tween, of course the vesicles stay whole and intact, so the antibody can reach just the outlayer surface of the vesicles.
Yep.
So if you add the detergent, the antibody can penetrate, so you can discriminate if the vesicles is present just on the membrane or in the inside.
Sorry, and that was not on the glass surface, so that you could have an idea about whether the mechanism 1 or mechanism 2 is preferentially there. That is not related
No,
to each other.
we cannot say it so
Okay,
far.
so those mechanisms still stand.
Yeah.
Okay.
Thank you.
No further questions? No, thanks. Thanks a lot.